Yeah, good morning, everybody. The title of my topic is actually the conclusion of the research I've conducted. Chromolaena is a serious invasive alien shrub. It's native to the Americas. We know by now that its natural habitat range stretches from northern Argentina to southern Florida. Its native range also encompasses the Central America, that is the Caribbean island. So basically, the impact of this weed is that it's too economically and ecologically significant to ignore. So it impacts on biodiversity, agriculture, as well as livelihood. The first biological control program against Chromolaena odorata started in the late 1960s and continued up to the late 1970s as well. It involves, that time, the Parauchaetes insulata depolluting mud was released. Unfortunately, this effort was not sustained. Presently, in South Africa, the first biological control program is still very sustainable. Started in 1988. To date, it's still very viable, sustainable, and it's ongoing as we speak. Basically, there are two different biotypes of this plant, and we have the Asian West African Chromolaena biotype, and we also have the Southern African Chromolaena biotype. Now, the Asian West African Chromolaena biotype is widespread in Asia, parts of Sub-Saharan Africa, including West Africa, Central Africa. Recently, it has been documented that the weed has been found in East Africa. Now, the Southern Africa is only present. The Southern Africa biotype is only present in Southern Africa. Recent studies by Patterson and Zacharias, 2013, is in press as we speak. They found out that they've been able to trace the origin of the South African biotype to Cuba and Mexico. So we now understand that the Southern African Chromolaena biotype emanated from Cuba and or Mexico and or Jamaica. So basically, this biotype, these two distinct biotypes can be differentiated based on their morphological characteristics and genetic attributes, as reported by Patterson and Zacharias 2013. They can also be differentiated based on aspects of their ecology, as reported by Kluge 1990 and Criticals 2005. Aspects of this ecology that relates to Southern African Chromolaena biotype is more fire prone and is also more cold tolerant, whereas the Asian West African biotype is less cold tolerant. Although the weed arrived South Africa, was first recorded in South Africa in 1940s. Is now present in Limpopo, Eastern Cape, KwaZulu-Natal, and Mpumalanga province. There has been speculations regarding the performance and the sources of biological control of Chromolaena in South Africa. Incompatibility of biological control agents with Southern African biotype has been speculated as a possible factor responsible for the limited, sources of biological control of Chromolaena in South Africa. So the reason for this anecdotal report may be as a result of the fact that the agent Parauchaetes insulata was collected in Florida, United States of America, on Chromolaena plant, on a disparate Chromolaena plant. You know, although the insect did establish at one site out of 30 release sites, this followed over 1.9 million individuals of P. insulata were released. So we only experienced establishment at just one site. So to date, as we speak, feed population of this insect remain low. So based on this fact, we theorize, or we hypothesize that P. insulata larvae will prefer Florida Chromolaena as opposed to Southern African Chromolaena. And we also hypothesize that larvae reared on Chromolaena, on Florida Chromolaena, should have higher fitness and performance than those reared on Southern African Chromolaena biotype. So basically, this is how the life cycle of the insect, look like. The eggs take under 25 degrees Celsius. The egg takes 4 to 6 days to hatch, and larva, period lasts between 16 and 26 days. I've also observed that pupa period lasts for between 10 and 14 days, whereas the adults can last up to, irrespective of sex, can last up to 12 days. So we conducted 4 fundamental or basic experiments. We look at the host's preference of this. We try to feed both biotype or both forms of Chromolaena. Herein, after I will refer to them as diet, not biotype. We conducted host preference tests, larva performance trial. We also looked at the reproductive life history trial. Leaf consumption trial was also conducted. So basically, in the host preference trial, what we did was to place both leaves on one petri dish, and on this we introduced 25 unfed larvae into the petri dish, and this constituted one replicate. We had 40 replicates for that, and we monitored the position of the insect after 24 hours and the following 48 hours. So in our larva performance trial, we recorded developmental time, pupa mass, growth rate, larva survival, and emergence. We also look at female sex ratio. We also calculated host suitability index according to following Moore 1976. So we also conduct series of experiments to ascertain the developmental, the reproductive life history of this very insect on both diets. We recorded female mating sussex. We also looked at achieved fecundity. We looked at egg hatchability, duration of egg laying. We also recorded longevity. We also recorded the oviposition commencement dates. So the leaf consumption trial, we basically followed each individual throughout the larval stage to pupation. So we're able to measure the leaf consumption or leaf area defoliated by a single individual. So this is how the experimental setup look like. We've been able to observe that over a decade after its introduction, P. insulata larvae still prepared Chromolaena Florida Chromolaena. Basically, we recorded high survivor on both diet. This implies there's no obvious influence of diet on survival of the insect. We also observed that diet had no effect on total development duration. So, and we record, it is instructive to note here that male and female development time did not differ. So, yeah, I'll briefly discuss this. In some, it is, yeah, we'll have to know that in some Lepidoptera, the emergence of males always occurred before females, and this has been described. Or this phenomenon is understood as protandric emergence. Basically, there are two advantages of this phenomenon. One is it maximizes copulation opportunities for a male, and two, it minimizes the pre-reproductive period for females because they emerge when most, when most males are available. So we think that the absence of protandry on both diets be a clear disadvantage for adults, although with constant and asynchronous inclusion of the insect and the feed, this is highly unlikely. But it will be catastrophic very catastrophic if this happened in the feed, and this would impair mating success and overall fitness in the feed population. We reckon that female pupa mass were heavier on South African Chromolaena diet for biotype. Growth rates also follow similar trend. It's also instructive to note here that female were more higher irrespective of on both diet biotype. So the fact that female growth rate is higher than that of male is classic in capital breeders, because male body weight is not often correlated with fitness as compared to female. Now this is a table showing the performance and fitness related traits of the insulata according to diet on which larvae developed. With the exception of adult longevity that were significantly higher for individuals that fed on Florida Chromolaena or Chromolaena from Florida. Mating success, egg hatchability, duration of laying days, achieved fecundity, oviposition, commencement dates, female sex ratio, and even post-suitability were not significantly different. So the significantly higher pupa mass of individuals that were fed on Southern African Chromolaena biotype did not influence female fecundity and other life history traits. So it has been documented that in active moths, male larvae sequester pyrrolizine alkaloids during feeding, and they use this in excess pheromones. Females, it has been reviewed in Column 2008 that female choose male based on this fact. It does not seem impossible that males that fed on Southern African Chromolaena biotype may secrete sex pheromones that do not turn on the Florida females. It was not part of our hypothesis, and we did not test this. We also found out that leaf consumption did not diet did not affect milk consumption, although female fed more than the male. Yeah, summarily, we want to conclude that our findings indicate that P. insulata larvae. Our findings indicate that P. insulata larvae preference for Florida Chromolaena plant did not have any obvious negative consequence on the reproductive performance and other fitness-related traits of the insect in the laboratory. Although we're not sure what will happen in the field, under field situation. Therefore, we want to conclude that biotype incompatibility may not be the reason or factor responsible for the limited sources and of all performance of this insect in South Africa. Thank you.